We wish everyone a life filled with health, happiness, love, peace, friendship, and success. Lilacs, though peaceful, can be affected by powdery mildew, a common plant disease. Even the wealthy crepe myrtle is not immune to the effects of powdery mildew. The healthy dogwood can also fall victim to powdery mildew, impacting its vitality. Lilacs can suffer from leaf spots, which mar their otherwise perfect appearance. Leaf blight is another issue that lilacs face, affecting their lush foliage. Today, we explore how molecular approaches can help analyze and identify plant diseases. Our focus is on molecular analysis and identification of pathogens in nursery crops. What pathogen causes lilac leaf blight, and how can we identify it using molecular methods? Are powdery mildew diseases caused by the same pathogen, and how can we analyze them? How can we distinguish and identify powdery mildew in dogwood using molecular approaches? Let's delve into identifying pathogens like lilac leaf blight using molecular techniques. The universal primer analysis involves 4 steps: DNA extraction, PCR analysis, sequence analysis, and similarity analysis. The first step in identifying plant pathogens is DNA extraction, crucial for further analysis. In our PCR analysis, we utilized universal primers to amplify specific DNA segments. The results show distinct bands, indicating successful amplification with primer pairs ITS1, ITS4 and ITS1F, ITS4, producing products of 570 BP and 608 BP respectively. The sequence analysis provides a detailed view of the DNA segments amplified from the PCR products using ITS1, ITS4 primers. This data is crucial for understanding genetic variations and identifying specific organisms. Through similarity analysis using blast search, we compared our sequences with those in GenBank. The results show a 100% identity match, confirming the accuracy of our primer sequences and their alignment with known genetic data. Specific primer analysis involves designing primers, testing them, and verifying their specificity. This process is essential for detecting pathogens accurately and ensuring the reliability of our molecular diagnostics. Primer design for the ITS region is a critical step in molecular analysis. We have developed specific sequences and reversal sequences to target the ITS region effectively, ensuring precise amplification and detection. Our analysis identified a specific primer for the Alternaria genus within the ITS region. The polymorphic band observed in all samples indicates that Alternaria is the pathogen, with a band size of 370 BP confirming its presence. We have identified specific primers for Alternaria alternata, with successful amplification results across various gene sequences. This confirms Alternaria alternata as a pathogen in lilac leaf blight disease. Alternaria alternata has been identified as a pathogen in lilac leaf blight disease. Further research is needed to determine if it is the primary pathogen and to explore other potential pathogens involved in the disease. Lilac leaf spot is associated with Pseudocercospora biramifera, as evidenced by the molecular analysis. Understanding the pathogens involved helps in developing targeted treatments for plant diseases. Powdery mildew diseases affect various plants, and it's crucial to determine if they are caused by the same pathogen. Molecular approaches help in analyzing and identifying the specific pathogens responsible. The molecular analysis and identification process for powdery mildew diseases involves its universal primer analysis, sequence analysis, and specific primer analysis, providing a comprehensive understanding of the pathogens involved. The universal primer analysis reveals distinct band sizes for different powdery mildew diseases, indicating successful amplification and providing insights into the genetic makeup of the pathogens. Sequence analysis has identified specific pathogens responsible for powdery mildew diseases, including Erysipelas pulchra and Uncynelliella australiana, based on sequence length and CG%. Specific primer analysis for Erysippe pulchra in dogwood powdery mildew reveals distinct polymorphic bands, confirming the presence of this pathogen in the samples tested. The analysis of specific primers for Uncinelliella australiana in crepe myrtle powdery mildew shows polymorphic bands, confirming its presence across various DNA groups and materials. Microsphaera syringae japonicae is identified through specific primer analysis, with polymorphic bands confirming its presence in lilac powdery mildew alongside other pathogens. Distinguishing and identifying powdery mildew pathogens in dogwood, such as Erysippe pulchra and Phylactinia guttata, requires molecular approaches to ensure accurate detection and treatment. Specific primers for Erysippe pulchra have been identified, with distinct bands confirming its presence in Cornus florida and other samples, aiding in precise pathogen identification. Phylactinia guttata is identified through specific primers, with distinct bands confirming its presence in various samples, providing insights into its role in powdery mildew diseases. The summary of molecular markers and specific primer pairs provides a comprehensive overview of the pathogens identified, including Erysippe pulchra and Phylactinia guttata, aiding in targeted disease management.